today we are going to go through the enzymes lab, demonstration of how the enzyme lab is done and how we are preparing the chemicals, how we are measuring the enzyme activity. So the enzymes are kept as a diluted form, and this is a glucose oxidase enzyme, and the substrate is the glucose, that is the sugar that we consume, is a 6-carbon sugar. And the enzyme glucose oxidase break down this in the presence of oxygen, and then it releases hydrogen peroxide, and we are trying to measure the activity of this hydrogen peroxide after its glucose oxidase work using the compounds called phenol and 4-amino antipyrine. So that produces a color. The more color there is, the more enzyme activity. Okay? The main objectives of the lab that we are doing today is to study the effect of concentration of the substrate, which is the glucose, on the activity of the enzyme, and then we are going to look at the effect of temperature on the activity of the enzyme, and then the effect of pH on the activity of the enzyme. So these are the main goals of this one. So to start with, I'm going to do the effect of substrate concentrations on the enzyme activity. So the first part of this experiment, we are going to study the effect of substrate concentration on the enzyme activity. So you want to be always careful when not to mix up the substrate with the enzyme activity, otherwise the reaction will start right away. Okay? So what I'm going to do is to label the cuvettes. I'm going to label the cuvettes based on the concentrations, but you can also label them, with a specific number, so that you know which one that you are using for which concentration. So the concentration is measured in milligram per deciliters. So we're going to go from 60 to 30 to 15, 7.5 and so on. So each time when you dilute the concentration, it is going to become half the concentration, but the dilution factor is 2. So to have this, you can use the formula V1 plus V2 over V1 is equal to dF, and then figure out what that V1 is, and then you already know the V2, which is a 1 ml. So in this case, it's a 2-fold dilution. You mix 1 part of the stock with 1 part of the water. But we are doing a serial dilution. In this case, you add the stock only once to the first cuvette, then you add water to each one of the other cuvettes, mix and move, and then discard the rest. Okay, that is what I'm going to show you, how to do that for serial dilution. So this is the stock, and here's this micropipetters. The micropipetter, you can set it up with the dial on the top, and you can change this. Now it reads 1000. That is going to measure 1000 microliters equal to 1 milliliters. So I'm going to use a blue tip for this micropipetter, and the blue tip protects the micropipetter from being contaminated, and that is a right way of using this. Also, whenever you're using the micropipetter, you want to keep it in an angle like this and use your thumb to, take the liquid in and to eject the liquid out. Don't ever do this. This is going to contaminate the micropipette. These are simple tips that you want to be careful about. Also, some students do this using their, pointer, which is not efficient, so you get tired soon. So here's a micropipetter, here's the glucose. You want to make sure you're adding the right amount, and I am going to, and also you don't want to leave this like in a jerky motion because it will, take air in. So I'm going to do this very gradually, and the liquid 1 milliliter is taken in. So now I put the glucose back on the ice. And now I'm going to add it to this first dilution, and I have 1 milliliter of glucose. So to this, I'm going to now eject this tip, and I can use this button to eject this into a waste beaker. Now I take a fresh tip. So this is something important. You want to change the tip as much as possible, and also wear gloves to avoid contamination of the enzyme or the reaction. Now I'm going to take distilled water. So this is how you do the dilution. 1 part enzyme, 1 part of water. So now the concentration becomes half, the dilution becomes twice or 2x. So now I mix the two together, I discard the tip, I put the micropipetter back on the stand, and that's also avoid any contamination. There are little small closures that you can put on these cuvettes, and these closures will help you, do this dilution by mixing this. And there are other types of a way of mixing this also by pipetting in and out, or if you're using a test tube, you can use a vortex machine to mix this. Now I have the first dilution done. So the next thing I have to do is to move 1 milliliter into the next cuvette. I'm going to add 1 ml water to each one of them now, so that it's easy to mix, move, mix, move, mix, move like that. So I'm going to add 1 ml water to each one of the other cuvettes, then it becomes a lot easier. Okay. So the other thing you may be able to do is, instead of using these closures, you can also use a paraffin. So that's like a waxy type of closure, and that also you can use to do this mix and move instead of this plastic closure. Okay. And I have done all the way up to 5 and then 6, and then I'm going to do the 7. Okay. So now I have added 1 ml water to each one of them, and then I'm going to take a fresh tip, and then I'm going to go to the first tube, which has 2 milliliters. I'm going to take 1 milliliter from this and move it to the next cuvette. Okay. So here, so this one I'm mixing. See, I can do this type of mixing also. It gives an even mix, or you can also close this cuvettes like we did before, and you can also invert this upside down and mix it. There are 2 ways you can do that. And the other method I said is through because this gets very tight, but you can remove that. Now I have to do the next milliliters, and so this is 2 ml. I'm taking 1 ml from this and go to the third one, and same way I am doing this pipetting in and out to mix this. So this has mixed evenly well, and now I take 1 ml from this and then go to the next cuvette. So this is what is referred to as a serial dilution. So you are doing this in a sequential manner, and the concentration at each stage becoming half, and the dilution is 2x. Okay. So we have the 4th concentration done, and I'm doing the 5th concentration and mixing it well. Now I go to the 6th concentration, and this also you have to make sure that you're mixing it well. Then go to the last one, the 7th concentration. So the concentration are becoming half and half, like 60 became 30, became 15, and 7.5 and so on. At the end, you want to take that and then you want to discard that last one into a liquid waste, and we can discard all these things later on, and also the tips. Okay? So now we have all this concentration done. Then only I'm going to add the enzyme to do the reaction. So now we have all the concentrations of the glucose the substrate made. Now we are going to test the concentrations effect on the enzyme activity. Before we even do that, we should have a blank to compare with. This is like a negative control or the placebo. So this is what will be used as a reference. For the blank, we need enzyme and plain water without any glucose involved. So I'm going to add just 1 milliliter of water into a blank cuvette, and then I'm going to add 0.7 milliliters of water. Okay? So the tip, I'm going to leave it for now here, just a plain water. Otherwise, I'll put it into the waste containers. Now I need to change the pipette setting to 700 microliters is what I need for the enzyme. So here I'm going to add 0.7 milliliters of the enzyme and to the water. So this should not react. This should not give any color. So that's what we are going to do. 0.7 ml of the enzyme. I'm going to leave it here and close the enzyme. Leave the enzyme on ice all the time. Only when you need it, you take it out. So now when I add the enzyme, it's going to mix with the water and there should be no color developing in this one. So even if I mix like this, there's no color developing in this. Okay? So now we are going to discard the tip, and I will put it into the I used a pipette tip here. So there we go. And now we are going to see how the calibration works, and there is a tissue paper called Kim White, and we are going to use that Kim White to wipe the sides of this and measure the absorbance. So this setup is called as the spectrophotometer, and this is a specific one called Vernier spectrophotometer. It's a simple, effective device where you can measure single wavelength and 2 wavelengths. So you can see the way this is set up. There is an arrow pointing, and this is the length of the light that you want to keep the cuvette in. So this spectrophotometer is connected to the monitoring device called as the LabQuest, and this LabQuest has the software to measure the absorbance of the glucose concentration and the enzyme, whatever we are trying to measure. So going to give you as how to use this LabQuest device. So there is a menu option. There's a stylus that you can select. You can select the sensors, and from this sensors you go into data collection, and once you are in the data collection, you want to make sure that the interval is set to 15 and the mode is set to time based and the duration is set to 120. So to change these things, simply you touch this and it will give you the option to enter, and you simply say 120 and you're done. Okay? So once you hit OK, it will come back to this. If you want to change the wavelength, you touch on the wavelength, just say select change wavelength, and now if you want 510 nanometers, then you enter that as a 510 nanometers. It's coming very close to 510 nanometers. Then to calibrate the water and the enzyme, again you can press the sensor and there is an option called calibrate, and once you press the calibrate, and this will go through the calibration of the spectrophotometer, and now you hit OK, and now it is going through a warm-up time, and since we have connected this for a while, it has been warmed up already, you can skip the warm-up, so now it will finish the calibration. So calibration is that it is zeroing in the absorbance for the water plus enzyme, and that's where calibration is completed. You hit OK. Now water plus enzyme is showing 0 absorbance. So that is how calibration is done. Now we are ready to start the enzyme one by one. We are going to do the concentration of the enzyme one by one, and we will know how the activity is measured. So now we have calibrated the spectrophotometer. Now we are going to measure the concentrations one by one with the enzyme and measure the activity of this enzyme for every 15 seconds. So you should have a timer set up, ready to go, whatever timer that you have, and then you measure that like every 15 seconds that it is going to show. Okay? So I'm going to add 0.7 milliliters of the enzyme to 1 milliliter of each of these concentration. So this is already set up to 0.7 mil, and I'm going to take the enzyme, make sure there are no air bubbles getting into the pipette. So leave the enzyme back on ice, leave the lid on. Then I'm going to take the concentration 1. You can go from concentration 7 all the way up to the concentration 1, or you can go from 1 through 7. So the thing, once I add the enzyme, the reaction is going to stop. So I need to start the timer, and that should be kind of ready to go. So I'm going to leave this oxidase, and I'm going to add this enzyme. So now it is mixing together, and that's my 0 time. As you can see, already the color starts to develop, and I'm going to take the blank out, and I'm putting this enzyme in, and there you go. So now you can see the absorbance changing from 0 time point to the 15 seconds time point, and I can measure that like every 15 seconds I enter it into the data table. So that is the main thing, that on 30, like every 15 seconds, you have to keep monitoring that, even though there is a timer, but you want to monitor from your timer as to how this 15 seconds work. And you enter the data as you go, 15 seconds, 30 seconds, 45 seconds, 60 seconds, and 75 seconds, 90 seconds, 105 seconds, and 120 seconds. So that is the observation that you need to be writing down as the enzyme reaction is going on. So you can have one person as a timekeeper, and you can start writing down this. I'm just showing you how the enzyme reaction is being measured by the spectrophotometer. Okay? So in addition, once you are done with this, you will see the enzyme reaction has completed. You can see that color. This is what this is measuring. I have taken it out of this, and this is what this has been measuring, and the enzyme reaction continues to go, that it is reaching 120 seconds. Then you can stop that. Then you can go to the second concentration and repeat the same thing and measure all the concentrations and write down all the numbers for every 15 seconds. Enzymes are made up of proteins mostly. There are some RNA that is also considered as an enzyme called ribozymes. With enzymes, the proteins have a three-dimensional shape that can be altered by changes in temperature, changes in pH, and hence that will affect the activity of the enzyme. So the objective of the next experiment that we are going to do is to see how the temperature affects the activity of the enzyme. So to prepare that, so I have to have the substrate ready and the enzyme ready at different temperatures, and then I'm going to equilibrate them so that they are in that particular temperature setting. So I'm going to label the QFs to different temperature settings. So the QFs are, these are the enzyme. I'm going to keep it at like a 0 degree, and the other one is going to, that is on ice, and then room temperature is normally about 23 degrees Celsius is the room temperature. And then we are going to also measure the activity at 37 degrees Celsius, which is like a body temperature. And then lastly we will do at a 65 degree Celsius, which is pretty, scalding, much hotter than a bath water temperature. So this is almost closer to a hot springs temperature of fun. So in each of these QFs, I'm going to add 0.7 milliliters of water, and then I'm going to add 0.3 milliliters of glucose. So that is to equilibrate the glucose and the water at different temperatures. So once I have this, and I'm also going to prepare, so the way you can easily use one hand, you don't need two people doing this, so you will get the hang of it. So I'm putting 0.7 milliliters in each microfuge tube. So these are also sometimes called Eppendorf tube or Epi tube, based on the company that makes these microfuge tubes. Okay. So now I have 0.7 milliliters of water filled in all these things, and I'm going to eject the tip. I'm going to get a fresh tip. So make sure whenever you change solution, you also change the tip. And the glucose is over here. You're always good to separate glucose from the enzyme glucose oxidase. So I'm going to change the settings to 0.3 milliliters. That's all we need to get 1 milliliters of this. So 0.3 mils of enzyme in each of them. No, glucose in each other, not enzyme. We will get to the enzyme next. Jumping ahead. So the glucose is the substrate. We are just getting it diluted, and you're just using the same tip. I'm not touching anything else. And now these tubes are ready to be equilibrated at different temperatures. Okay. So now I want to get the enzyme also prepared the same way. So I will get 4 fresh cuvettes. So in this case, I'm going to label them as 0 degree Celsius, and the same way we did the labeling as 23 degrees Celsius, and the third one is the 37 degrees Celsius, and then the fourth one is 65 degrees Celsius. Okay, so we have all these ones ready to go. So now we have to take the enzyme and also have them equilibrated at different temperatures. So I have labeled one cuvette for 0 degree, and then I'm going to take 0.7 milliliters of enzyme into that 0 degree cuvette, and I have a closure for this cuvette, and I want to make sure I am using a fresh tip separately for enzyme, separately for the glucose. So that is the main thing that you have to keep track of. So long as you are dealing with a clean cuvette and a clean tip and not touching the substrate, you should be fine. You can use the same tip for each one of them. So we have the 0 and 23 completed. Now this is 37, and then I have the 65 degree 0.7 milliliters. So we will have all of these closed properly and equilibrated properly. So what I'm going to now do is to use these closures, these plastic closures, to close so that there's no water or anything that will get in when I am going to keep it inside those different temperature conditions. Okay, so this is the second one, and now you have all these closures done, and there should be one more closure. Clear this, and there you go. So the 0, I'm going to keep it on ice. So this is how the enzyme as well as the substrate I'm going to equilibrate on ice, so they get used to that conditions. And then I have the 23 degrees Celsius, which is a room temperature, and this 23 degrees Celsius, I'm going to leave it at the room temperature right here. So the 37 as well as the 65, I had to take it to the incubator. So these are called as the water baths. So these are temperature incubators. one on the left side is set at 37 degrees, one on the right side is set at 65 degrees. So I take this, the substrate and the enzyme, I put it on a float, which will keep it intact and also stay above the water. So I take this float and then open this lid, and then I leave it inside so that it is exposed to that temperature. So that is the main idea of this float being used here. And there are also different type of floats. This is another type of floats. So long as this can go in there, and this will also have to go in. This is, you have to make space for that. And this is a 65 degree, it's kind of really hard, so you got to be careful when you leave it in there and when you take it out. Okay? So now this is equilibrating at this temperature for about 5 minutes. Then we are going to take one sample at a time and conduct the enzyme activity measurement, just like we did before. By mixing the substrate with the enzyme, the reaction will start. Then I put it on the spectrophotometer, and then we'll start the experiment. Okay, so let's go back to the spectrophotometer and see how that is done. So you don't have to come back and forth into this incubator when the measurement is going on, because after 5 minutes of exposure to a high temperature, the enzyme changes the shape and it is going to be inactive or active depending on the optimal temperature of the enzyme. So now we are going to test one temperature setting and its effect on the enzyme activity. So now I have a cuvette that was stored at 0 degree and it's already equilibrated. So I have to open the lid, and it's kind of tight, and I take the glucose that was also equilibrated at 0 degrees. So now I have to add the glucose into the enzyme. The reaction is going to stop. Then I am going to measure it with a spectrophotometer, which was already calibrated, so I don't need to redo that again. So I'm just taking this glucose and water already equilibrated. I'm just adding to the enzyme, and I'm just using the same tip because I'm going to discard this tube. I don't need this anymore, and discard this tip. Now I can just close this very quickly. I can mix this, otherwise I can do that with the pipette also. So here it is ready to go, and you can also wipe with the Kim wipe. Now this is measuring the absorbance, and I can also set the timer. Somebody is setting up a timer, and every 15 seconds that you measure that, how the absorbance is increasing. So each of them, that based on the temperature, now it's a 15 seconds is done, you write down this absorbance. Then you wait for the next 30 seconds, then you write down this absorbance, and each 15 seconds that you're writing down. The same way, once you're completed with the measurements of 0 degrees, then you go into the room temperature one. Then after you finish with the room temperature one, you go into the 37 degree, you complete that. You don't need to be going back and forth into the temperature setting. And then last is the 65 degrees, then you complete it, then you get all the observations done. Okay, now the third part of this experiment, we are going to test the effect of pH on the enzyme activity. As you know, pH is a measure of hydrogen ion concentration. As the pH goes up, the hydrogen ion concentration decreases, and the pH goes down, the hydrogen ion concentration increases. So the hydrogen ion concentration can impact the 3D shape of the enzyme and then the activity of the enzyme. So here we have 4 different buffers of different pH conditions. So we are going to test how each of these buffer will affect the activity of the enzyme. So I'm going to use a pH paper to test what is the pH of these ones. So I'm going to keep the pH paper one for each of these buffer, and then I'm going to put a little bit of buffer on each one of them and find out what is the actual pH. And pH papers are normally very approximate, so these are not actual accurate as much as a pH meter, but for this purpose it just works fine because there's significant differences in them. Okay? So what I'm going to do, you can use either a transfer pipette like this or a disposable pipette tip. In this case, I'm going to use a disposable pipette tip, and I'm going to take this buffer 1 and see what the pH of that buffer. I'm going to take just a little bit of that and then put it on the pH paper, and then I'll discard the tip over here, and so I'll just let this sit on for a while. Then I'm going to go to the next buffer, and I'll leave this open for now and take a little bit of that and leave it on this pH paper, and then discard that. Then go to the third one and test the pH of that. Discard it and leave it over here, and then the last one you're going to test. So as you can see that the pH paper is changing colors, and we just match it. This is more called a colorimetric measurements. So, but the pH meters, they work based on electrical conductivity. So that's how these are all like a calibrated based on each one of them. So by looking at this, color chart, you can tell, you can see whether this is matching any of these. These are like the way you want to hold it like this, and so this will match closer to this one. Okay, so that is what this is about. pH 3. That's an acidic pH range. So you can mark this as the pH 3 here. Okay, and the next one you want to check what is the color of this and see which one. There are two sides to this, the 1 through 0 to 7 and 7 to 14. So this doesn't match any of these here. It's pretty close to pH 5 is what I would call it, and otherwise you start to see that green color start to show up, and you can call this as the pH 5. Okay. And let's go to the third one. So the third one is closer to pH 7. So pH 7 is over here, as well as pH 7 is over here. That's the closest that you can get. So let's call that as the pH 7. Let's go to the last one, which is so you look at that. It's all these kind of you can see the dark blue color start to show up. It's closer to pH 10 based on the color combination you can find, and that is the pH 10. So as you can see from this, the pH is increasing in this order. This is pH 3, 10 to the negative 3 molar hydrogen ion, 10 to the negative 5, 10 to the negative 7, 10 to the negative 10 molar hydrogen ion. Now I'm going to take a sample from each one of them, and I'm going to close them next. Okay, so the amount that I'm going to put is about 0.7 milliliters of this buffer. So this is so I have the cuvette 1, and I have putting 0.7, and then I'm going to write the pH of that as pH 3. Okay, so that is what I'm going to keep it here, and then the next one I'm going to label it as the pH 5 over here, and then I have the pH 7 and the pH 10. So all these cuvettes are ready. Now I'm going to add 0.7 milliliters. I'm going to change the tip each time I go to a new solution, and this is pH 5. Let's close this, and that goes into the pH 5. Let's close that. And then next is the pH 7, goes into the pH 7, and then comes the pH 10. Close that. So after you measure in the 0.7 milliliters, now you have to add glucose to each one of them, and before that you close all these, make sure they don't spill on the table. Okay? So now I would add glucose to these pH so that the enzyme has something to work on. Okay? So here's the glucose. So I'm going to do 0.3 milliliters of glucose. So that will bring the total volume to 1 milliliters. Okay, there you go, 0.3 milliliters, and each of these, there we go. And I can also add without touching this, but I would rather put it in and then I can mix like this. You can also mix using a lid, and I'm going to discard that. I'm going to go to the next one. I'm going to have the 0.3 milliliters to the next pH. I'm going to go mix back and forth, and discard that. Then the third one, discard that. Then comes the last one. So now I've prepared all the glucose solutions in different pH buffer conditions. Okay. So now the goal is to see how the pH of the aqueous solution is affecting the enzyme activity. That's an extra thing you want to look at. So to do this, you can take one pH at a time, and then you're going to use a spectrophotometer like we did before, and take the enzyme, and I have just make sure that you have the fresh tip taken, and I'm going to do 0.7 milliliters of the enzyme. Okay. So this is set up to 0.7. There you go. So 0.7 milliliters of the enzyme, I'm going to add only one at a time. Don't add to all of them, then all the reactions will stop. So I'm going to start with the pH, this is pH 5, and then pH 3 is right here, okay, the pH 3, and I'm going to add it to this pH 3, then I'm going to put it into the spectrophotometer and measure the absorbance. Let's just get to the spectrum. So now I added this, and then this is the acidic pH, I mixed it, now my timer starts, okay. So I'm going to stick it in here and set up the timer. So I can, this should have, be ready, but you always have somebody doing the timer, and it is already measuring this, and the concentration is increasing as now it's about, 15 seconds when it comes, and you measure at 30 seconds, and you measure at 45 seconds, and so on. So you do these measurements up to 1. Now this is at the acidic pH. You can see that activity is different, and once you complete this up to 120 seconds, go into the second pH 5, and then pH 7, and then the pH 10, and you write down all the observations. Okay? So that would complete this lab. Now you have tested the effect of concentration of substrate, the effect of temperature on the enzyme, and then effect of pH on this. So the first one was effect of substrate concentration on enzyme activity. The second one is effect of temperature on the enzyme activity. The third one we are doing now is the effect of pH on the enzyme activity. So once you have all the data, you want to complete the analysis part by entering that information in Excel, and we will provide you sample data to do the analysis.